membranes, transport, gradients, all super important components of how cells work. So when we left off on Friday, we were talking about what specific molecules make up a plasma membrane. Now we want to understand a little bit more about how it works. So we used this term before saying that a cell is selectively permeable because we want important things to enter. We want any molecules that are the middle of a sort of pathway to be able to stay inside the cell. We don't want them to be leaking out, but we do want waste products to exit the cell. So the way we do that is by altering the composition of our plasma membrane. And we're going to talk about that today. But first we want to understand what can get through the cell membrane in the first place on its own. So we saw before that our plasma membrane is made up of phospholipids, right? So phospholipids have a polar head, both on the extracellular portion, so the outside, and the intracellular portion, the inside of the cell. Then in the center of this bilayer, we have fatty acid tails, basically doubled up. So we have a really thick nonpolar region. That's what makes the main barrier for things that are trying to get across the membrane as a whole. So very small things and very uncharged things are able to dissolve directly across the membrane, to diffuse directly across. So we think of these things also as being lipid soluble because they mix well with lipids, right? So we're going to have small uncharged molecules. And in particular, we're going to be interested in gases. So oxygen and carbon dioxide are two very important gases for cellular functions. So we'll be able to see that they are able to dissolve directly across that membrane. So they're going to diffuse directly across. Then we have sort of gradations in the middle. So we'll see that water and urea, the membrane is moderately permeable to them. So they can get across, but it's a little harder than a tiny oxygen molecule. Larger things and polar things really have trouble. So polar organic molecules, which is specifically going to be sugars, right? For example, we saw those had a lot of oxygen, making a lot of polar functional groups, right? They're going to be repelled by the membrane because of this non-polar layer in the center. So they might be attracted to it. They might try to get across, but they're going to bounce off these fatty acid tails. They can't squeeze through. Okay. Ions have complete charges, right? They don't just have these little charges accumulating due to electronegativity. They actually have full electrons added or missing. So they're not going to get across. They have even lower permeability than our sugars. Big things are also going to have a lot of trouble getting across. Other things that are polar, other things that are charged. So if I were to draw a line between like what the membrane is permeable to and what it's non-permeable to, I draw it kind of in the middle here, right? Between this moderate permeability group and this low permeability. While I'm saying I would draw something, why don't I actually draw it? Okay. So let's think of the membrane as being permeable to these things and not permeable to these things. Okay. Okay, so that's our basic division. There are gradations depending on the chemical structure, but we can group them kind of like this. Okay. So a reason we want to know what the membrane is permeable to is we're going to be focusing a lot on how things cross the membrane, which direction they go, and how they get there. So we're going to be thinking about two concepts. Today, we're going to be thinking about passive transport, which is the movement of molecules across the membrane without the use of energy. And then on Wednesday, we'll be thinking about active transport. So we're going to add in movement of molecules across the membrane where we do need a source of energy. Now, in order to identify whether something is undergoing passive or active transport, we have to start by understanding something called a gradient. Okay. So in this class, what we're going to focus on is chemical gradients, although as you move forward in your courses, you'll see that the big picture is actually a bit more complicated than that. for right now, we're thinking about chemical gradients. So what a gradient is basically a difference in concentration across a membrane. So a difference in how tightly packed molecules are. So we're going to see the first thing we want to do when we're trying to figure out passive versus active transport. So we want to look at our cell, look at our membrane. We want to basically count up how much of a molecule is on the outside of the cell versus how much is inside of the cell. So in this case with the purple ones, right, we'd count a lot of purple molecules here outside the cell. This is a depiction of a high concentration outside the cell. We don't have that many purple molecules on the inside, so low concentration inside the cell. And we can see this in these other situations as well, right? Here in the second image, we'd see we have a higher concentration outside the cell. If we think of the outside of the cell as the top, a lower concentration inside the cell. And in this third image, we'd see that we have a high concentration inside the cell, so lots of these purple particles, and a low concentration outside the cell. So this is our first step, is identifying the gradient. When we think about passive transport, things are going to move down the gradient. So by down, we mean from high to low. So from high concentration to low concentration. This is a natural process. This is just how molecules want to move in the world due to laws of thermodynamics, which we are not going to get into. But this is what doesn't require energy. If you move from high to low and everything's just trying to reach equilibrium and spread out evenly on the inside and the outside, that doesn't require energy and therefore is passive transport, which is our topic for today. Okay. Now we can use specific terms to talk about these gradients when we're talking about a cell. Okay. So these gradients build up in the first place, partially due to selective permeability. Right? The reason you can have a lot of something outside the cell and only a little inside the cell is because certain things aren't able to cross the membrane. So we can control things like that. We can concentrate stuff if we block stuff from moving. Okay. So that's useful sometimes because living cells have to maintain a pretty constant internal environment. Right? We need to tightly control what's inside the cell because we want that environment to stay relatively stable so that we know how our chemical processes are going to work. We know what's inside. Lots of chemical reactions will be dependent upon concentrations. So it's important that we don't just let whatever is in the environment in. Okay. So when we see a difference in concentration, lots of something outside, only a little inside, or the reverse, we call that a transmembrane gradient, meaning across the membrane, we have different concentrations. Okay. As we start to think about what those concentrations are, we talk about relative tonicity. Now, these next three terms we're going to talk about are ways of describing that transmembrane gradient. And they refer to specific types of gradients. So what we want to focus on here on this slide, we're thinking about the amount of solute, which is the thing that's dissolved, in the solvent, which is water. Okay. When something is isotonic, this prefix iso means like the same. So a cell that is in an isotonic environment, that means we have the same concentration of the solute outside as we have inside. So if we look at how these are spaced, they're spaced pretty evenly outside versus inside. So that is an isotonic solution because it's the same as the inside of our cell. Okay. We have two words to refer to gradients where we actually have differences. Okay. So if we have a cell in a hypertonic environment, what we're saying is that there's more of the solute in the environment than there is on the inside of the cell. Okay. So hyper means like more extra, right? So there's more or extra solute dissolved in that water outside the cell compared to the amount of that solute dissolved inside of the cell. So that's hypertonic. Hypotonic is the reverse. Hypo means low or little. So if we have a cell in a hypotonic environment, that means we're in an environment where we have a low amount of that solute outside, which means that relatively we have a lot of that solute dissolved in the water inside the cell. All right, so we have three terms. We have isotonic, referring to the same concentration inside and outside. We have hypertonic, which refers to a lot of the solute outside the cell. We have hypotonic, which refers to only a little of that solute outside the cell. Okay. So those terms are referring to the environment outside the cell as compared to the inside of the cell. Okay. In passive transport, we're going to have a couple of types, but remember, none of them are requiring energy. That's what makes them passive, right? If you're just passive, you're just lying there, you're not expending any energy. Okay. So the simplest type is diffusion. So diffusion is going to happen basically with those small nonpolar molecules that interact with lipids, right? They can get directly across that membrane. So they just dissolve, diffuse directly across the cell membrane. So our examples here are oxygen, carbon dioxide. We had that on the first image of a membrane. We'll also see that lipid hormones can dissolve across membranes as well. Okay. So diffusion is always passive. Specifically, if we're going through our steps, right, the first thing we want to look at is what are our gradients? Because this is passive transport, it's always going to be movement from an area of high concentration to an area of low concentration. It's diffusion if it's just directly across the membrane. Now keep in mind with transport, total movement is going to stop once we equalize our concentrations. So once we have that isotonic situation, we're at equilibrium and movement will stop. Okay. So basically what we have when we're talking about diffusion, think of this dashed line as our membrane. We have a concentration. We then just spread out those dots. We'd see that some moved into the cell, right? So on net, we went, right, from super concentrated. We had some move over here to where we had a low concentration, right? Because here we have high. Here we have low. We had movement until they were equally spaced. Okay, so that's diffusion. A special case for passive transport is going to happen when we have a gradient, right? So we have a difference in concentration outside and inside the cell. So in this case, we see a hypertonic solution. So we would identify that there's a lot of this solute outside the cell and only a little inside the cell. But we have lots of molecules that can't get directly across the membrane, right? They can't just do simple diffusion. So easy diffusion, simple diffusion directly across that membrane. But they still want to reach equilibrium. We still want those concentrations to even out. So what we're going to have happen in this case is something called osmosis. So osmosis is the diffusion of water across a membrane. And it's pretty neat. So the cell membrane is permeable to water. And there are things that make the cell membrane more permeable to water than the basic components that we've talked about thus far, but we're not going to get into those details in this class either. But for now, just think about cell membranes as permeable to water. Water is always going to move down its own gradient. So water is going to move from where there's lots of water to where there is less water. And this is the thing that can trip people up sometimes. So if we're looking at this hypertonic solution, what we see is lots of solute outside and a little solute inside. So I want you to all think in your minds for a second, where are we going to have more water? And draw some in. So we're thinking about this solute concentration, right? The solute is basically taking up space, right? These are molecules, they take up room, they're physical, they exist in the world. So we're going to think of like, oh, I can draw circles. Cool. Okay. So we're going to think about water as filling in the room that the solutes are not occupying, right? So think of each of these blue dots as a water molecule. Okay. So on the inside of this cell, we have lots of room for water. On the outside of the cell, though, solutes are taking up lots of room. So there's not that much room for our water molecules. So if we were to think about this hypertonic solution, right, it has high solute concentration, low water concentration. And we don't usually think about concentrations of water, but basically they are the inverse of each other because of these spatial relationships. So on the inside of the cell, right, we have low solute concentration, high water concentration. So if this solute is able to get across, it would move down its gradient, right? This would not be osmosis if we have the high solute move towards the low solute concentration. But sometimes we have our selectively permeable membrane, so that can't happen, right? If that can't happen, in order to equalize these concentrations, we're going to have water move from where we have lots of water to where we don't have very much water. And this will essentially be watering down that solute. So we can think of high solute concentrations as kind of pulling on water, right? We can think of this density of solute as pulling on the water from the inside of the cell. So when we're thinking about osmosis, you kind of need to flip the solute kind of concentration in your mind. We're identifying the gradient, but then we want to remember that water is going to be basically the reverse of the solute. Okay. So this is important to cells because it actually has a physical, visible effect on cells. If you remove water from a cell, it gets smaller. If you add water to a cell, right, it still has to be inside the membrane. So that cell is going to swell and bulge. And if you put too much water in, it's even going to burst. So here we see a red blood cell, right? If you place it in a hypertonic solution, hypertonic means lots of solute, not much water. So the water is going to move from the inside of the cell to that environment, trying to water down that environment because those solutes are pulling on the water. So these cells are going to undergo crenation, which is the shrinkage of the cell due to that hypertonic solution. Now the reverse, we call osmotic lysis, which is the swelling and even the bursting of a cell due to too much water entering it. So that's going to happen if you put a cell in a hypotonic solution. Like, imagine you put your cell in a glass of perfectly pure water, right? Distilled water, you have nothing but water molecules in there. You still have solutes on the inside of your cell, right? If they can't leave the cell, you'll have water move in, trying to water them down. It's going to keep going in, keep going in, keep going in, because nothing fixes that gradient if there's really nothing in the water, until there's so much water on the inside that physically the membrane can't hold it anymore, and then it would burst. So that's osmotic lysis. Now, we're not going to focus on plants much, but they act the same way. The only difference is that they have a cell wall in a plant cell. So instead of bursting in a hypotonic solution, they're just going to become really firm, really crispy as pressure pushes out on those walls. And instead of crenation, where we have like our blood cells shriveling up, they don't shrivel in quite that same craggy way because of those cell walls, but the cell walls are going to dip in. This is basically what's happening when something wilts, right? We have water leaving to join this hypertonic environment, so this cell is going to get floppy and wilt. Okay. So plasmolysis is wilting, and turgor pressure is the pressure that makes a plant cell firm in a hypotonic solution. Okay. So don't worry about these exact words. I think I'm going to make sure that those aren't on the test, but do understand this concept of being able to tell if you're creating pressure from this water inside the cell versus if you're wilting because we have water leaving the cell. Okay. So next we have facilitated passive transport. Okay. So this is still passive transport. So we'd still identify a gradient. In this case, facilitated passive transport, we have things moving from high concentration to low concentration. And they're able to do this because of a protein with a channel. So this is directly pulling off of your problem set from plasma membranes on Friday, right? So we have a protein embedded in the plasma membrane. So it's a transmembrane integral protein. It has hydrophobic regions and hydrophilic regions, right? Specifically, it has hydrophilic regions, so like polar stuff, lining this channel so that polar molecules and ions can travel through from the outside to the inside. So they can't diffuse directly across the plasma membrane. They can't do simple diffusion because they bounce away from this nonpolar region. But once we open up this channel and line it with polar R groups, it can now go through this channel. So facilitated transport is just things diffusing down their membrane, but through a protein. So it's simple if it's directly across the lipid bilayer. It's facilitated if we need a protein for it. But otherwise, they're the same, high to low. So if we look at a channel protein, just to understand that a little more, a channel protein is really kind of just like a pore in the cell membrane. It's a protein that creates this tunnel, right? In the future, you will talk about channel proteins that are gated. Don't worry about them for this class. Just assume that I'm not showing you gated channels. But you should know in the future that cells can turn these channels on and off, basically. We're going to stop here and we're going to continue with transport and carrier proteins on Wednesday as we start to talk about active transport, because these are going to be really crucial to the process of active transport.